Hi everyone, I am Gobika MP. Today I would like to detail the topic factors influence microbial growth. For ecological studies, bacteria are usually cultivated and studied under laboratory conditions. So, the growth of the bacteria have different factors, mainly the nutritional factors and the physical factors. The nutritional factors also include the numerous media, etc. The physical conditions are the pH, gaseous environment, temperature. So, nutritional and environmental are main factors influencing the microbial growth. So, the successful cultivation of bacteria require a awareness of all of these factors. So, first we see the nutritional requirements. All organisms require a source of energy. So, in the all living form, there is a source of energy. The source of energy is the chemical energy. They are considered as chemotrophs, and the source of energy is the radiant energy. They are called phototrophs. So, second nutritional requirement is the all organisms require a source of electron for their metabolism. Organisms use inorganic compound as electron. They are called lithotrophs. So, mainly photolithotrophs and chemilithotrophs. Then organisms use organic compound as electron donors and are called organotrophs. Then third point is all organisms require carbon in some form for use synthesizing cell components. So autotrophs require high source of carbon. Then next point is all organisms require nitrogen in the form of cell components. Bacteria can use atmospheric nitrogen, and also they are needed for the nitrogen for their formation of cellular components. All organisms require sulfur, oxygen and phosphorus for cell components. All organisms require metal ions for their normal growth. Then all organisms contain vitamins and vitamin-like compound. Last nutrition requirement is the water. So their body 70% or 80% is fully made up of water. These are the nutritional requirements. So the very important topic, nutritional types of bacteria. There are mainly 4 types of bacteria based on their nutrition. First, phototrophs. Second, chemotrophs. Third, autotrophs. And the fourth, heterotrophs. First, we explain the phototrophs. So, phototrophic bacteria, their source of energy is the radiant energy. Then they use inorganic compounds as electron donor. They consider as photolithotrophs. They use organic compound as the electron donor. They are called photoorganotrophs. Mainly, the example is Chromatium ocani uses H2S as electron donor, oxidizing into elemental sulfur. Second type of nutritional types bacteria is the chemotrophs. Here, the chemotrophic bacteria, their source of energy is the chemical substance. So, they use organic compounds as their electronic donors. They are called chemi-organotrophs, whereas the inorganic compound, they are called chemi-lithotrophs or chemi-organic compound, they are called chemi-organotrophs. Example, the bacteria Nitrosomonas use ammonia as their electron source, obtaining energy by oxidizing ammonia to nitrate. Next is autotrophs. Autotroph organisms that can use carbon dioxide as the sole source of carbon for assimilation are termed autotrophs. Example is the Nitrosomonas species. Last or mainly the last type of is the heterotrophs. So heterotrophic bacteria have been studied more extensively that it is the disease-causing bacteria present in human, or there is the infection-causing organism. So heterotrophs, they are carbon source in the form of inorganic compounds. So all the disease-causing bacterium or disease-causing microbe is the example of heterotrophs. Then last type is the obligate parasite. So parasitism mainly here occur. So obligate parasites and bacteria have not been successfully cultivated in artificial medium, and their nutritional and physical requirements are not understood. So they are depend on the living organism. Example, Mycobacterium tuberculosis causes tuberculosis. So next is the bacteriological media. So we culture the bacterium in the laboratory condition. They need a bacteriological media. Their mainly compounds are peptones, meat extract or yeast extract. There are mainly different types of media. First is selective media. Second is differential media, and third is assay media. Selective media is selection of the microbes, and differential media, certain reagent when incorporated into culture media may allow differentiation of various kinds of bacteria. So they depend on the differentiation of microbe. Then assay media is used to the assay of vitamins, amino acids and antibiotics. So it is also used for testing of disinfectants. Next is media for enumeration of bacteria. So they are help to enumeration. Next, media for characterization of bacteria. Then next is maintenance media, and the last one is the solid semi-solid media. Next is preparation media. So preparation of media mainly in these components are mixed together and undergo autoclaving. Then they are used as a bacteriological media. Next is physical condition. So physical conditions are mainly depends on the culture of microorganism. First is temperature. So temperature, all of the microorganisms they have optimum temperature. Under the influence of temperature, there are mainly three type of microbes: psychrophiles, mesophiles and thermophiles. Psychrophiles, they are live or they are cultured at 0 degrees Celsius or lesser than the minimum or optical temperature. Mesophiles grow best within the temperature range of approximately 25 to 4 degrees Celsius. So, all of the disease causing bacteria in the human body is the mesophiles. The last one is the thermophiles. They are culture or they are become live when the temperature of above the 45 degrees Celsius. Next physical condition is the gaseous requirement. So based on the gas or gaseous condition, there are mainly four types of bacteria: aerobic bacteria, anaerobic bacteria, facultative anaerobic bacteria, and last microaerobic bacteria. Aerobic bacteria require oxygen, anaerobic bacteria require not oxygen, they are live without the oxygen. Then facultative anaerobic bacteria, they are become facultatively anaerobic. Then microaerobic bacteria, they have no any influence of oxygen, but they are live in the without the atmosphere. Next, last is the acidity and alkalinity. So acidity, they have a minimum pH. So the pH of the media mainly 6.5 to 7.45. So we prepare in the media, we check the pH for the culturing of microbes. Then apart from this, miscellaneous physical requirements is also contained, such as illumination of light, so source of energy, and also the hydrostatic pressure. Thank you.